We've started, good to go.
Okay,
It's recording, you're live.
excellent. Take
You're done it.
1 hour.
Maybe I can pick up the laptop.
Sure. Yes, I think
I
you
did
started. That's
this time.
weird.
You're good to go.
Perfect, thank you. Let me just close. Yep. Okay. All right, so we are going to get started. I have a lot to say, so I'll try and do it in a bit of a structured way. So for those of you who I think, I'm not seeing a ton of new faces, but my name's Lauren. I'm a pharmacologist and clinical trialist at the Georgian Fee Center for Healthcare Innovation. I'm also the chair of this C.C.R.P Winnipeg chapter, along with Brendan and Tarpin, who is our dedicated secretary. He really does most of the most of the work. So I wanted to talk about outcomes. I think they're a really, really important part of designing a clinical trial, and I'm going to tell you about them today. I took off the drug studies. I'm a pharmacologist. I kind of lean towards the drug study examples, but really this information applies to studies looking at devices, looking at nutritional products, looking at any kind of intervention. So I tried to take that drug focus off a little bit. So again, I had a lot to say, but I thought I'd focus on kind of 3 key areas, which is selecting appropriate outcomes, reproducible outcomes, and outcomes that have clinical relevance. And so I sort of structured the talk to talk a little bit about why I think outcomes are so important and why I thought it would be a good use of our hour today. What guidance is out there? So what do we know? Where are there tools we can access? How do we get information on, what we should be doing and best practices? We'll talk about selecting, what's appropriate for selection, reproducibility. So that's going to talk about outcome measurement, so measuring things in a consistent way, as well as a bit about reporting guidelines. What does clinically meaningful mean? How do we, clinically meaningful to whom? And I'll end off with a couple of tips and a couple of examples, and then a little very quick summary. So what are outcomes? Outcomes are really how we measure safety, how we measure benefit, how we measure harm. Outcome is what's going to tell us whether something is beneficial, whether something is inferior, whether something is equivalent to what our control arm is. So these can be reported in a variety of different ways. They include biomarkers and lab values. They can be patient-reported outcomes or PROs, and we'll talk a little bit more about those. They can be reported by children or teachers or parents or clinicians or nurses or social workers. They can be medical devices. They can be apps that track things. There's lots of different, the term outcome, is super variable, and what is an outcome can also be variable. So why do outcomes matter? Well, I come at this talk a little bit from a systematic reviewer perspective. So my job at the end of the day to take these 3 or 4 clinical trials that have been published in pediatrics, these are often very small studies, and try and make some practice recommendations based on, a breadth of evidence. So one clinical trial, if anyone has an example, I've asked this question many times, will never change practice. Generally, even regulatory requirements are for at least 2 studies. And this is because we need to compare and combine and really figure out what treatments work best under what conditions. So what patients are likely to benefit. Obviously, there are different clinical care settings that we need to think about. And so in order for studies to be replicated, we have to be able to reproduce their findings. So we need to look at different populations, again, usually like a different clinical environment. We don't generally do the same 2 trials identically in under identical conditions. And outcomes are really important. They tell us how big our study is going to need to be. If you are doing a real power calculation, not just sort of working backwards from how much money you have, which, as Rob just walked in the room, I'll tell you this probably happens more than it should. But outcomes are important. They're going to tell us, what that difference is, and we'll talk about that. And they also contribute to research waste. So this is a pretty famous paper that came out in 2014 looking at research waste. And they estimated that 85% of research, or $220 billion, has been wasted because of inadequate production and reporting of clinical research. And you can see where outcomes play a role. So you can see in the beginning phase, when we're designing our question, when we're figuring out what the decision is, what the question is we're going to ask, If we're not assessing important outcomes, that contributes to research waste. And when we get to the end of the spectrum, when we look at reports and trial reporting, more than 50% of planned study outcomes are not reported. Those are not reported because they're not sexy, they're not significant, they're, somebody found something that was going to garner maybe some media attention. We'll talk a little bit about outcome reporting and outcome reporting bias. But I also think that outcomes play an important role when we think about bias, when we think about statistical power, and when we think about replication. So if I don't know how you measured your outcome, how am I supposed to replicate that? So as I mentioned, one clinical trial isn't likely to change practice. So clinical trials and evidence go through this sort of knowledge to action cycle where we look at multiple clinical trials. We do evidence synthesis. So we look at systematic reviews and meta-analysis. We use those to develop clinical practice guidelines, which then get championed and endorsed by our key opinion leaders. Then they can be, disseminated and shared, and eventually some change management will occur. And that's what we're doing. When we're doing clinical trials, our end goal is really here in yellow. No matter what kind of study you're doing, the goal is the same. It's to improve outcomes for children's families and the healthcare system. So, no matter where your trial is, if it's early phase, late phase, the long-term picture is really the same. The goal is to improve outcomes. But this can't happen if we can't identify enough information about the clinical trials to feed into evidence. I say this again from my systematic reviewer hat. If I can't find your trial, if there's not enough information published in your trial, it's not registered, if there's no way for me to access that information, I can't include it in the clinical practice guideline. So this is just a very quick example to frame sort of why this is important from a patient perspective. So this is a person, very industry person who has a question about asthma, says, does this new treatment work better for my current treatment? So the systematic reviewer goes to your computer and says, okay, what RCTs have been done on this new treatment? So in this case, we find 3 studies. So one, a study with children with severe asthma, another study in severe asthma, it's not necessarily clear what the population is, and a third study in children, but excluding children that are over a certain BMI. Again, this is a hypothetical example. So then what we ideally would be able to do is look at these 3 studies and say, does the treatment work? Who does it work best for? What conditions, were they more likely to see benefits? What were the benefits? What were the risks? So there's a couple problems here. So the first question is, what do you mean by work better? So work better for me may be very different than work better for Dylan or work better for Brendan. So in this case, they say fewer trips to the emergency room. I don't want to go to the emerge for my asthma. I want it well controlled. Well, trial 1 measured pressure in the airways, trial 2 measured mortality, and trial 3 measured quality of life. So we can't actually pool those studies. We can't actually do a meta-analysis. We can't actually compare them because they're not measuring the same thing. And so this is also sort of related to the concept of outcome reporting bias. So there's a few ways that this happens. And basically what it means is that you're picking and choosing, you're kind of cherry picking what outcomes get presented in that publication. And remember, that publication is a systematic reviewer's first source of information. So we try many times to contact authors for more info. I think about 5% of the time do we ever get responses for more information. We try and find protocols, we try and find registries, but these are not always available. And so this happens, I'm sure this isn't news to you all, and it happens in 3 different ways. So the first is a subset of study outcomes. So you measure 25 outcomes, you have these huge long case report forms, but then you go to write up your report and highlight 3 that maybe happen to show the biggest difference. You could be selective reporting of a specific outcome. So let's say I measure blood pressure at 5 time points. There's no difference until time point 3, which seems to show a difference. So I only report blood pressure at time point 3. Or incomplete reporting of a specific outcome. Again, that could be a similar example, is really cherry picking what information you're going to present in the report. And a recent systematic review that was published in the BMJ in 2014 found that 86% of studies had some form of outcome reporting bias. So this is another study by Anwen Chen, who is the sort of godfather of SPIRIT. So Anwen Chen is the lead author for the SPIRIT guidelines, which are about clinical trial protocols. And this was some work that they did underpinning the need for actual reporting guidelines for clinical trial protocols. And so this was a study. They looked at 76 clinical trials. Again, this is back in 2004, and they looked at any changes. These were where they had a protocol available. So they looked at changes from the protocol to the report in what outcomes they measured, what outcomes they described. Some were just completely omitted, some were non-primary, some were just not mentioned at all. But overall, 62% of their trials had some discrepancy in primary outcome reporting. But this is really a simple concept. Avoid outcome reporting bias, register your trials, publish your protocols, and just be honest. Be transparent. If there were changes that were made, these should be justified. There are lots of reasons that would be justifiable to change an outcome. Maybe patients thought it was too burdensome, or there was some reason. We just want to know what that reason was. It should be clear that there were changes and clear why the changes were made. It's simple as that. So what help is available? So I thought I'd start with help and then go into a bit more of the specifics instead of ending with it. And there are lots of different kinds of help that are available. There are hundreds of methods papers out there. There are core outcome sets. There's guidance, tons of different guidance from the regulators. There are reporting guidelines. And then, there are your friendly folks at CHI across the street that are happy to help. So methods papers, these are a bit, different presentation. Right. So this slide actually changes to add, librarians are really your best friend here because they can be a little bit difficult to find. There's not necessarily great search terms for methods papers. So they can take a little bit of time to dig through to identify them. And lots of times these are population and intervention specific. So I'll show you what I mean by that in just a second. But one thing that I thought was really important is when you do find these papers that are specific to the kind of research that you're doing, save them somewhere so that when a new person is hired or a new trainee is starting, you have some of those methods papers that they can read from the beginning to get an idea of good practice and areas that they can do, areas where they can improve their research. And so this is what I mean by they tend to be population specific. So these are some of a selected group of my methods papers that are all in pediatrics, all about clinical trials. I think there's one surgery paper in there, but they're very specific. So they tend to be hard to find in the vast sea of all of the papers that are available in PubMed. So I would recommend a librarian being definitely a great resource for helping you find some of those in your disease areas. The second thing is core outcome sets, and I'm going to give an actual example of one of the core outcome sets that we've developed we are just finishing up the publication for right now. So core outcome sets, these are disease specific, and they are a minimum set of outcomes to be reported in any clinical trial or even for quality improvement. And I say a minimum for a reason. I say a minimum, but they tend to be not that small. So I have seen them range from about 5 to 13 outcomes, but they're not there to tell you you must only measure these items. They do still advocate that whatever your research question is, your primary outcome should be selected to match to be able to answer that question. So if your question is about cost, and none of the core outcome set items are about cost, well, that should still be your primary outcome. So it's not telling you only what to measure, but it's trying to set a standard where if all studies measure the same things, then at least on these important items, we can compare and contrast between studies. And so it increases interpretation, and it allows people like me who have that task at the end of the day of evidence synthesis, it allows us to combine and compare studies. There's a fabulous resource. So there are over 1,216 ongoing or previously completed core outcome sets. There's a lot of them. If you are working in an area, there is probably one somehow related. It might be in an older population or a slightly different, maybe disease severity. But in general, they're great resources. So the COMET, or the Core Outcome Measures and Effectiveness Trials data set, they actually have a core resource pack. So if you do decide that you want to develop a core outcome set, and I'll give you an example of how we did that, there's guidelines that are available. There are actually guidelines for making these guidelines. So there are standard methods that you have to go through. So you can't just sit in a room with 4 people and say, what do you think is important? What do you think is important? Okay, core outcome set. They're quite rigorous, and it has taken, about 3 years of work. So these are not to be taken lightly. And I think if there is one that is available in your disease area, they absolutely should be implemented. So take a look if you haven't already. commoninitiative.org. Like I said, you can just keyword search it. It's super user friendly, and it'll tell you if they're still in development. So you might find a new cool research team to collaborate with. It's a global initiative. So I thought I'd talk a little bit about our core outcome set. So we have just finished, we had our consensus meeting at the end of April, a core outcome set for neonatal opioid withdrawal syndrome. So neonatal opioid withdrawal syndrome is a bit of a hot topic the last few years because of the increase in pregnancy. And in opioid withdrawal syndrome, it's almost similar to what you would expect in adults. If you discontinue their opioid exposure, many will go into withdrawal. So if a child is exposed in utero throughout pregnancy to opioids, at birth when that exposure is discontinued, they develop withdrawal syndrome or opioid withdrawal. Now, not all babies develop withdrawal. Some are managed non-pharmacologically with breastfeeding, rooming in, dim light, lots of cuddling. And some do require pharmacotherapy with either one or multiple medications. So it's a disease that has a lot of variability. Don't know a ton about it. There's almost certainly a genetic component, but we really need more research. And so this is what led us, and this is, I won't go into my whole background story, but this is an area where I was working in my Ph.D. and really was unclear as to how outcomes would translate across sites. And I'll give you an example of that coming up. But this was clearly an important area where we needed to do more research. Every 25 minutes, this is actually down to 12 minutes now in the latest version of this, which I meant to change, but time. This data comes from the NIH, and you can see that the number of cases of opioid exposure in pregnancy is increasing, the number of children with withdrawal is increasing, and the costs associated with management are high. This is just some Canadian data. Again, just to highlight that this is a condition that no one really thought too much about in the research space. Maybe 15 years ago, there were so few cases it was actually considered a rare disease. And since we have again seen such a dramatic increase in the number of cases, we actually aren't able to classify it as a rare condition anymore. So we were unable to do some of those surveillance projects that require a certain incidence rate. Because we've seen such a dramatic increase. So this is, we have now over 1,000 cases a year in Ontario, which is a 16-fold increase from 2002. So this is our methods, and I think this is a pretty standard method with one small difference based on our population. So to develop a core outcome set, you start with a systematic review. Anyone who's done a systematic review knows that's probably about a year's worth of work, depending on how much literature there is. And we use the systematic review for 2 reasons: to identify outcomes that have been reported and to identify participants to participate in Adelphi. Adelphi is really a fancy term for a series of surveys that's with the goal of a consensus. So it's a consensus process where the first round is open-ended. So the participants say what outcomes are important to you when you're managing these children, and they can write in whatever they like. We combine all those outcomes with all the outcomes from the systematic review, and all the outcomes identified by parents. So in general, when you're doing a core outcome set, you would include persons with lived experience in the Delphi. Our population is very research averse. Our population does not want to complete 3 surveys online every few months that are very long and lengthy. It's a very different and challenging population from a research perspective. So we felt that actually one-on-one interviews would be more meaningful to them and a better way to engage with them to really even explain what a core outcome set is and why we're doing this research. So we actually did our interviews separately, but again, in most cases, you would include them in the Delphi. So the second round of the Delphi is prioritization. So you score an outcome from 1 to 9 on how much it would influence your care. And then Delphi round 3, you get presented with all of the scores, and it's basically a reduction process. Do you feel it should be in a core outcome set? Yes, no, not my expertise. And then that goes to a consensus meeting. So we set consensus definitions that are published in that protocol. Again, like I said, we just had ours at the end of April, which goes into creating a core outcome set and doing some feasibility testing, and of course publishing all that with lots of details. So in this example, we actually had 63 potential outcomes that were discussed. So this included 28 outcomes from a systematic review, 55 outcomes from a Delphi, 18 outcomes that were identified by parents, and then of course there's some overlap in what those outcomes were. So there are 63, don't take any pictures of this, not publicly available yet. And just to show, these are what our parents identified as important. And we actually plan to do 20 of these interviews, and after a year and a half and 6 interviews, we said that's going to have to be enough. And that was simply based on we weren't identifying any new themes in the last 2 interviews, which generally if you're doing a qualitative kind of study and you're not identifying any new themes, you can kind of sort of hypothesize if you've reached saturation, if you will. But what was really important is that all 6 parents independently said that the duration until adequate symptom control, which was not called that in the interviews, it was how long until they stop shaking in my arms, how long until they stop crying, how long until they can be consoled. They want that treatment to act fast. Not one clinician, so our Delphi participants were clinicians, nurses, and social workers, not one of them identified that as an outcome that was important to them, which we thought was really, I didn't really know what to think about it, to be perfectly honest with you, as I thought, has nobody asked these women what was important to them? And the answer was honestly no, they had not. And so this outcome did make it into our core outcome set. This is just to give you an idea of what outcomes we're looking at. Is consolability, difficulty feeding, it doesn't matter what the outcomes actually are. But really they're 9 outcomes, but it's really 10, 11, 12, it's really 13 outcomes. So it's a lot of information. And this started from 63 outcomes, and there were many discussions around what would go in there. So we're in the point now where we're trying to harmonize definitions. What does a difficulty feeding look like? What does parent-infant bonding look like? How do you measure parent-infant bonding? These are all questions that we are still discussing now, and we'll go into the development of our explanation document. So in terms of what other help is available, there are regulatory guidelines. There's lots of regulatory information out there. They do promote the use of core outcome sets, so we do have representation in our core outcome sets from members from the FDA as well as the European Medicines Agency. This is so that they're aware that the core outcome sets are happening. They're aware of the evidence that went into informing these core outcome sets from an international perspective. They also have lots of documents on extrapolation and modeling. So if you're like me and you work in pediatrics, there are criteria for when you can model some outcomes and what information is needed on the disease. To do that, there's guidance on pharmacokinetic studies. The FDA also has a table of biomarkers that have been validated as outcomes, and that's all available online. So there's lots of things that are available to be searched. And there's also industry regulatory academic partnerships. So I'm a part of the International Neonatal Consortia, where we actually have been working on this opioid withdrawal core outcome set, and we just presented it to the FDA at the beginning of May. And they're developing master protocols. So their goal is for some of these more specialized disease areas where they haven't seen a ton of drug development, like neonatology, to develop master protocols and to actually have these discussions beforehand with the clinical community and the families as to what outcomes are important to them. They also have extensive and I think really great guidance on patient-reported outcome measures. So a lot of times, maybe people aren't sure about the use of PROs in drug development studies, so actually studies that are being used for marketing authorization. And they have really, I think it's quite comprehensive documents. I think I have a little bit more on that here, because I think this is a really great resource for those of you who are using patient-reported outcomes. So this is what the FDA is looking for. They're looking for how many things are you asking, what's the conceptual framework, why are you asking these items, what the condition is, the population, how the data is being collected, how it's being administered, what the response options are. So you always have to have a response for I don't want to answer this, or it doesn't apply to me, those types of things. Recall period is a big one, and they have extensive guidance on if you're asking people about the last 2 weeks or the last 2 months or the last 6 years or when you were pregnant 25 years ago. So they have different criteria for recall. Scoring, weighting, format, the burden on the respondent, which I think is really something we need to consider. I just reviewed a survey that was like 42 pages, and I went, you think people are going to do this in your waiting room? You've got to be kidding me. Why are you asking, this many questions when what you really want to know is this? And so they do consider that, and their guidance, I think, is actually quite good for these instruments. Oh, I forgot that I put this in here. So they actually even have a framework for how to develop patient-reported outcomes and how to test them, how to confirm that they are actually reflective of clinical care and what, isn't just important to me, it's important to a diverse population. And so they have, again, how to prepare your protocols, the involvements of statistical analysis plans, and all of that, which is readily available online. So if you are interested in that, do check it out. Reporting guidelines, I hope most of you in this room have heard of SPIRIT and CONSORT. These shouldn't be new concepts. These are basically your baseline reporting guidelines for clinical trial reports, which is CONSORT, and clinical trial protocols, which is SPIRIT. And they're there to help guide the writing of a clinical trial report so that when people like me find them, there's enough information there for us to be able to answer our questions. And they increase the ability to replicate studies and inform practice guidelines. There are 413 reporting guidelines. I told you guys, there's a lot to talk about. There's a lot of tools that are out there, and almost too many. If I had to pick a side of not enough or too many, I veer towards too many. And so you can find reporting guidelines for very specific types of studies. So if you're doing a study within a trial, or a SWOT study, or an N of 1 study, or a crossover, multi-site study, there are specific reporting guidelines probably for you. So again, this is something that I recommend to check out. It has search and free text, so you can type in either a disease variable, or you can type in your study design method and see what's available to you. And I think it's really important that you think about this when you are designing your study, because if you're looking for them as you're writing up your study, but you didn't do 10 of the 20 things when you were designing your study, it's kind of hard to really have the same impact as you would if you had this at the beginning and you were designing your study with all these considerations in mind. So it is something that I recommend checking out before you select your outcomes. There is a plug-in, if you will, coming for Tidier and CONSORT that is specific to outcome reporting. So there are a couple items in SPIRIT and CONSORT already that are outcome related, but they're not quite complete in the sense that you'd have enough information to replicate that trial. So Anwen Chen and David Mower, who are the authors of SPIRIT and CONSORT, and this is a project that's being led out of my former postdoc lab with Martin Fringa and SickKids, and we actually had. Oh, there it is. So we actually just had our consensus meeting in April. So these are sort of the reporting guideline dream team, I call them. And so these will be available soon, and they'll be implemented in the next iteration of the SPIRIT and CONSORT extensions as well. Help at CHI. So I just want to spend a little bit of time here because we can't do any of these things for you. But what we can do, these are all great things that are important, is we can support your staff to do these kinds of things. So if you are budgeting in a grant and you want to do a core outcome set, that's fabulous. We're happy to provide the method support. We're happy to provide the training that's required. We may even be able to, help with some software or some analytics, but we can't do a literature review for you. We can't make a core outcome set for you. As I mentioned, our core outcome set is like on year 3. So, these are big projects, but what we can do is help provide the method support. So we have, great patient engagement that can help you set up some of those focus groups. So a lot of times that patient engagement piece to a core outcome set might include a focus group. So, we can help with that. So, like I said, we're happy to help from a methods perspective, and I think it's good to utilize us if you are bringing trainees in to do this kind of work. We're happy to help with that as well. So now on to the real stuff. And I mean, I'm sure this first statement is really probably one I don't have to say, but it's really important that when you select something, it's not just because you can measure it easily or it is cost effective to measure or you think CIHR peer review panels are going to like it. It should really be what matters to doctors, to participants, and to families. And I think it's a really thoughtful discussion that maybe sometimes gets kind of swept under the rug as like, oh, well, this trial did it this way, so we're just going to do it that way too. And I think it really requires more discussion than that. And I think it's really important that we involve more than just our clinical team in our little silo that's been doing the same thing, the same way for 30 years. And especially when you're choosing your primary outcome. So your primary outcome is especially important. This is going to be chosen to answer your question of interest. Again, it's going to be how you base your power calculations and your analysis. And it should reflect a meaningful change. So what is a meaningful change? I'll talk about the minimum clinically important difference in a second, but this often comes up as, we have these fabulous oncologists and they want to look at a specific protein expressed, in a tumor biopsy. And you ask a patient and they say, I don't care about that protein. That means nothing to me. I care that I can go to work. I care that I can sleep at night. I care that I'm not vomiting 6 times a day. And so I think there's sometimes a little bit of disconnect between what is objectively measurable and what is actually meaningful to those who live with the disease. So the minimum clinically important difference, or the MCID, is how different you think the outcome will be from the control arm. So, for example, you think your new treatment is going to be superior by 20% compared to standard of care, or you think it will be non-inferior by 5%. So when you do a power calculation, you actually have to set a bar for what your difference is going to be in that primary outcome. And you can't just pick numbers like 20% or 5%. You actually need to provide some justification for why that difference is important. So why is 20% important? Is it because if they're 20% better at this time point, they're less likely to have an emerge visit down the road? You need to really think about what you're trying to measure and how and what that important difference is. Now, these are ways, these are discussions that you have to have before you meet with your statistician. So when you think about these margins, we'll call them, so whether it be a margin of superiority or inferiority or what the same looks like, because the same is never going to be the same down to 10 decimal places, right? So even when we're doing equivalent studies, we have to have a margin for what the same really means. So always, be conservative, align your study design with the question you're trying to answer, and provide rationale. So if you pick 20%, you need to have some kind of justification for that value. So don't just throw, 5% is improvement or 50% is improvement without any reason as to why that 50% matters, what it means for long-term outcome. So reproducibility, we have to replicate studies. Again, I think this is the 4th time I'm going to say it, is that, not one trial is going to change the world. And if we're trying to improve healthcare and health systems, we have to be able to replicate studies. And most trial data isn't shared openly yet. I hope that there's a movement and I hope that it will change and maybe there'll be some more of this moving forward. But the only access people who write clinical practice guidelines have is what's published in the medical literature. So whether that be in a registry, in a protocol, or in a report, we can't reproduce what we don't know. So if it's not published, we can't reproduce it because we don't even know that it happened, or we don't know what the methods were. And really, I just want to talk about 2 things with reproducibility. Reporting guidelines I talked about before. I think these are really, step 1 for reproducibility. Make sure you're reporting all the information that's required for us to reproduce your studies. And the second topic is about the measurement of an outcome, because I think this is where a lot of reproducibility tends to get bogged up, is in how the outcome is actually measured. So if you're measuring something as simple as weight, are you measuring weight at birth, weight at discharge, weight gain, weight loss, weight in pounds, weight in grams? Some of those things can be converted, obviously, back and forth, but you can't necessarily compare discharge weight with the number of days to regain birth weight. They aren't necessarily outcomes that we can compare. So I thought I'd spend a little bit of time on measurement. So the first question is really, is there a tool? Is there a tool that's been validated in your population? If you're trying to do a 6-minute walk test in newborns, well, that doesn't really make much sense, right? Does it? Even that's a standard of care, or a standard tool for measuring, ability. Well, it has to be matched with the population. And training is a big one. So a lot of times we see multi-site studies, and in one site we have a very experienced nurse coordinator who has spent 20 years of service in the NICU and knows how to reliably evaluate criteria X. At site 2, we have a trainee that's just been hired who's a master's student that the PI has seconded to do these interviews with patients basically when they have time. And so you have to think about who is measuring the outcome, if it is something that there may be some variability in where a sensor is placed or how that measurement happens, and what training is required for that person to be able to reproducibly measure that outcome, right? So if I measure the same outcome in the same participant, it should be, very similar, if not the same. You don't want something where I measure it one day and I measure it the next day and they're totally different, or if I measure it and then Shelley measures it, that they're totally different, right? So another thing that I sometimes see is, are the same measurement tools being used? So some of the studies that I have been involved in, they're actually using different versions of the same equipment across different sites. This means different calibration processes. This means different standard operating procedures. It's not to say that this is bad or can't be done, but it does need to have some thought around what are the implications. Do these machines function at the same capacity? Do they have the same limit of detection? Do they have the same sensitivity? Is the tool generalizable? Is this like some high-tech outcome tool that is never going to be used in clinical care? Well, you might want to think about that before you're creating this magical thing that isn't necessarily going to be in practice. Thinking about calibration and operation procedures, do you have a trained tech that has been doing maintenance on the same machine for 15 years? Or is this something where you've never done maintenance because you never thought you had to? Happens, I promise you. And you don't have operation procedures in place. So it's really important that across sites within the same study, there are standard operating procedures for how outcomes are measured and for the measurement tools that are being used. And I would always like to see some kind of measure of retest reliability, test retest, and that means consistency, in myself measuring the same thing, and consistency between Shelley and I, who may be at different sites measuring the same thing. So it's something to definitely think about, and I don't think we probably do as good a job as we can on that. So this also actually comes from the patient-reported instrument guideline from the FDA. But I think it's really, it's actually just very usable and friendly way to think about measurement properties. I don't know about you, but I find measurement properties not to be the most exciting part of my day. But I think this is a good job of laying out test, retest reliability, what the validity is, construct versus content validity. And again, this is available on the FDA website. And I won't spend too much more time on that, but definitely something to think about, especially if you're developing your own scale. So if you're developing a new quality of life scale for a specific, disease condition, this is a really great resource to look at what the measurement properties are. So the last R, I think it was relevance. Relevance was my last R, reproducibility and reporting. So ask patients and families what matters to them. It's honestly as simple as that, but it's not, because it also needs to be done in a rigorous way. So I think sometimes, it really varies into the level of engagement that investigators will have with the patient community. Some investigators, again, like a core outcome set where they've formally been engaged and there's, documented process of their engagement is one way, but there are oftentimes investigators sit down with a few patients and they say, what matters to you? And it's not to say that that's bad. It's maybe not so reproducible, but it is at least a start at the discussion of what matters to them. Look at the literature, that's going to be your best source of information for what other studies have shown in terms of differences between treatment and control. So when you're justifying that 20% difference or 5% difference, you should provide references as to, where why you think that's important and where that's coming from. Of course, core outcome sets in general, must include engagement with families or people with lived experiences. So these are great tools that have already asked people what is important to them on usually an international perspective. And our surrogate endpoints and biomarkers important to participants. So I think, again, this is sort of goes back to that example in oncology where, you may be so excited that their blood levels of XYZ are down 5%, but they're actually more nauseous and not sleeping. It may not be what matters to them. So again, when we think about surrogate endpoints, these are being used more and more frequently in drug development because basically a surrogate endpoint means you can measure it at a time point, but it will correlate with what we see in 5 years or 10 years because measuring, disease-free survival at 10 years, well, that takes a long time. So surrogate endpoints, again, we just want to make sure that they do have some relevance to the participants. So a couple of tips and then just a couple, just two, I think, examples of think about who is actually going to measure this outcome and where. Sometimes, in pediatrics, we have teachers measuring outcomes. Teachers are very busy. If you've ever seen a room full of kids, then you understand that they may not have the most amount of attention to pay to a 45-page questionnaire. Maybe like 5 or 6 questions is probably all they're going to be able to get through, in a busy day. So thinking about who's measuring the outcome and where are they measuring it, the training that's required, and this is really important, I think, also across sites. We do want to see some consistency in how those people are trained. And of course, with GCP, you should be documenting all of your training requirements, looking at what resources are required, whether that be equipment or, a checklist or a measuring tape. Are they all using the same measuring tape? Little things like that, you should try and think about ahead of time. Also, if you're expecting there to be some difference, so if it's a subjective outcome score, so in neonatal opioid withdrawal, one of the criteria for scoring is a high-pitched cry. Well, if you're a busy NICU nurse on hour 14 of a busy triplet delivery, let me tell you what that high-pitched cry is probably going to score a little bit higher than if you are fresh out of bed and you had a great date night last night. It's not the same objectivity that, say, a blood biomarker would have. So if you do have subjective outcomes, that's fine. But you really need to think about, am I scoring that high-pitched cry? There are actually audio whatever thingy tools that you can actually measure and they will score it for you. Something like that might be a little bit more reproducible than just asking a nurse or almost worse, a parent to score that item. So again, just thinking about what the reliability is and if measurement is consistent across sites, a lot of that has to do with what infrastructure is in place. And population considerations, and that's, can't go through a whole talk without talking about pediatrics. Oh, okay, we're going to do pediatrics first. So outcomes in pediatrics, a little bit of a challenge, a little bit of an extra challenge, I should say, because there's a lot of variability in standard of care. So standard clinical outcomes are not measured the same across provinces. And differences in how they're treated may actually contribute to these outcomes. So we need to be clear, it also matches with standard of care, right? So we have to think about the standard of care when we're thinking about maybe differences in outcomes that we see across sites. We don't have great age-validated measurement tools for most outcomes. Don't have really great reference ranges, especially if we're talking about NICU and preterm babies. We don't have great validation for biomarkers. And the practicality of patient-reported measures at school really needs to be thought about because a lot of times, especially with some of the attention and hyperactivity disorder studies, they're like, oh, well, the teacher at school is going to measure this child on day 5, day 7, day 9, day 12, day 13, day 30, day 45, day 60. And it's not really practical. And so, with kids, we need to think about, with the intervention, we need to think about, does it taste okay and all of that things, and will the kids actually take it? But with the outcomes, we also need to think about, is this something that is practical for the family to measure? So if you need 5 data points, don't ask them to do 45 different things. So the other example I want to talk about is length of stay in hospital. And this relates to the neonatal opioid withdrawal outcome set that I presented because the top outcome in the systematic review was length of stay in hospital. Anyone have a guess as to why that might be? pass marks. Cost, yes, because it's easy to measure. Because you can do a retrospective study and it's very clear how long someone was in the hospital, right? There's a discharge day, there's a birthday. That's something that's easy to extract. Not necessarily the best outcome. So in neonatal opioid withdrawal, I come from Ontario, where we used to do at-home weaning. So we used to send children that were stabilized home to finish their weans under the care of a nurse and a pharmacist. These aren't just like kids going home without any care in place. Here in Manitoba, we don't have the resources to do that, which means our babies stay in hospital until their wean is completed. So the length of stay in hospital is much longer for a child here in Manitoba than it might be for that same child, same family, same situation in Ontario, because they'd be weaning at home. So I can't compare length of stay in hospital. It means nothing about how long they were on treatment, how much treatment was required, the severity of their diseases. It really honestly tells me nothing. What it does tell me is here in Manitoba, if we're following, kids in the same hospital with the same discharge policies, that might give us a little bit of insight as to who's, costing maybe more or less money. But we really also, length of stay is influenced by many factors, particularly in this population, especially if we're waiting on social worker foster parents. This can take some time, and often the children will stay in hospital. Their weaning is completed. They're no longer on treatment for withdrawal, but they're waiting for social work or a foster family or some other reason. So it doesn't necessarily reflect the disease severity at all. Counting days, are we talking about days in intensive care? Are we talking about days in hospital at all? Days in the NICU, days in the birthing unit, the new women's hospital, we're going to be able to room in. So we'll hopefully see a lot less admissions to the NICU. But again, if I'm comparing to a program in BC, where every mother rooms in, the hospitals are designed to keep the women in hospital. It's not, I wouldn't even call it a hospital, it's more a care clinic where they stay as long as they would like to stay. So they stay way longer than they do here in Manitoba, not because the kids are sick, but because the families require support and they choose to stay in hospital longer, center care longer. So, it's not really a great outcome to compare treatment across sites. But it is easy to extract. It's easy to extract from charts. And it helps for a cost estimate. Because at the end of the day, we should be including health technology assessments into our trials. We should be thinking about what the cost effectiveness is, because no one's going to implement an intervention that's going to cost a lot and not deliver, in terms of improvements in outcomes. So just an example of an outcome that is commonly reported. It was the most commonly reported, and some of the challenges that occur. So I think I'm wrapping up the last little bit that I wanted to say, which is really that it's a critical, it almost might be the most important, this and choosing your control arm are really some of the most important decisions that you're going to make when you're designing a study. And I really advocate for involving multiple people, preferentially outside of just your little clinical treatment team when you are selecting these outcomes. And I will say from a peer review perspective, it's one of the first topics that will come up for discussion. If that primary outcome is not justified or justifiable, you're not going to get any money to do your study. There's lots of guidance that's available from reporting guidelines, regulatory guidance, methods, papers, experts. There are people who are experts in outcome reporting. And they're a resource that are available and something that you can think about. It's free. There's lots, and at CHI again, we're happy to help. So if you do have a study that you're thinking about designing and you just haven't even a discussion, these are free 1-hour discussions. We might be able to point out some tools that you're not aware of, or even just ask questions that you might not have thought about in terms of when you're kind of framing your question and thinking about what outcome might be best to answer it. And just the last point, I think for the 5th time, outcome selection is important. We need to be able to reproduce these studies for them to have impact. So, we can't reproduce what we don't know. So just be very clear when you're writing up your studies and provide us with as much. So, I think that's it for me. I actually have no idea what time it is. Some time for questions. So it is 12:52. So does anyone have any questions or anything they'd like to talk about. Anyone had these kind of troubles comparing outcomes across studies or trying to maintain a standard across multiple sites? All your sites are just amazing and do the same thing the same way at every turn? Well, you.
Selecting the right tools.
And that also highlights the importance of analysis is if you are going to have these cutoffs where over 25 is normal and under 25 is not normal, well, what is the relevance of 25? You know, these are things you need to think about, and you need to actually, you may have it in your brain where this is relevant because, but you actually have to put it on paper for other people to look at your work and understand why you made these decisions. So I think that's okay, I think we are done for the summer. I'm looking at Tarpon. Yeah, no. Okay, so we have no one scheduled for June. We do have someone scheduled for September. Unfortunately, this room has been booked up for classes. So I think we are back in Chown in September in the 4th floor in 474 where we used to have fringe rounds. We will try our best to highlight when the room changes. We're going to try and book a bunch of rooms in advance so that hopefully we can try and stay in the same place. It proves a little bit more challenging than maybe it should. Lunchtime is popular for booking rooms. So just have a great summer. And if you have ideas for rounds, stuff you want to talk about, speakers you want to recommend, Turpin is your man. Shoot he or I an email and we can get them scheduled. Happy summer. Right up, Turpin. I think I'm stopping this Turpin, but I'm honestly not sure. Yeah. Yeah,
not the one photo.
Just the core outcome set. That's the only thing that can't go out.
If you have the photo of the thing, I just won't put the data.
Oh yeah, the protocol. Yeah. That's fine. The methods part is fine. That's all. That's published in our protocol.